Okay, now we continue. As people do chromatin interaction maps, they observed 3 levels of interactions. The very first level is loops. So this region is interacting more with another region. And so on this high-C map, you can see here, so basically, you are looking at a region. So the lower boundary here is just, in the chromosome. You can see that there are many red things here, meaning that when things are very close by, they do interact a lot with each other. But if you look at this on the top of this triangle, it means that's the interaction between the left corner and the right corner. And you do see the color getting lighter and lighter. It means that as things become far apart, their interactions decrease. However, occasionally you would see an enrichment of these dots in here. That means there are more interaction between this, the dotted line here on the left and the dotted line on the right. There are more interactions. So this is a more specifically enriched interaction. And if you analyze the data well, normalize the data well, as we mentioned in the previous part, you will be able to see these type of specific interactions. These appear as really enriched dots. But as we mentioned, this depends on the experiments, and restriction enzyme you use and how deep you sequence. They are seeing some of these dots, but not a whole lot. What they do see a lot is these kind of domains, which are triangles. You can see within the triangle, things look pretty red, but between the triangles, it's really quite white. And so what they call this are topologically associating domains, which are these triangles in the high-C map. And so usually these are self-interacting regions, roughly 100 to maybe 200 kb triangles. And there are a lot of interactions with each other. And you can see here that there are a lot more interactions of regions within, but not as many interactions without. So we can look at whether the genes are really regulated together or not. And so this is, so for example, if you look at the H3K27 acetylation ChIP-seq data, and just look at, for example, by distance, if you look at this left 100 kb and the right 100 kb and look at how far apart they are, and look at the overall H3K27 acetylation signals between the left and right. If you look at things that are within the same TAD versus across the different TAD, across the different triangles and within the same triangles, you do see something like this on the right side. And so, so the left and the right side, even though they are the same, for example, if two things are 50 kb away, but if they are in the same TAD, their K27 acetylation signals are still more correlated. But if they are the same 50 kb away, but cross two different TADs, their K27 acetylation signals are much, much lower, suggesting again that things within the TAD have more interaction with each other, and the genes within the TAD are usually more likely to be correlated. So in some sense, it's like a little module where things can be regulated together. And so interestingly, what people found is that these type of TADs are quite conserved within different cell types. You look at the high-C data in different human tissues, they are conserved. And in this particular example, we're even showing that this is a region that's present in both the human genome and the mouse genome. In different species, you can still see the TAD, these kind of triangles to be conserved. So the TADs are even conserved between species. And so you kind of want to know, can we identify what are these regions or little modules that genes are likely to be co-regulated or at least have more interactions with each other. This is in one of the very earlier high-C papers. This is 2012, where they look at the interaction map. They generated something called a directionality index, which is to ask, for example, at each of the locations, you ask, because we will always sequence the high C with paradigm sequencing. So with each location, you can ask, where is the other sequence read in? And so for example, if you're looking at this location, can you guys see my arrow in here? Within this location, there are things that interact with it from before, but there are also things that interact with it from after. And because when you do the sequencing, you can map the read in this location, but then you can also ask what is the other read, where does it resides in? And so in this case, it's using a 40 KB bin to ask, that's one bin, what all the reads that lands in this 40 KB, where it's the mate pair, the other read, is the read in the left or in the right? And when they look at this, they notice something interesting. If you look at this location, most of the interactions it has are with things after it. And so it's kind of more biased in its directionality. And so you can see it's like mostly red, it's all positive. All the reads are things after it. Whereas if you look at, for example, this region as the anchor, most of the interactions happened in front of it. And so if you look at the other read, it's all negative, it's before it, right? So the green is all negative. And so if you build this interaction matrix, whereas if you look at a region around here, there are interactions before, there are also interactions after. And so that's kind of close to zero. And what they found is that if you look at overall this type of phenomenon, by dividing the genome into 40 KB bins, you will see that this region is mostly positive, and then it will be close to zero because it's interacting with both ends. And then after a while, it will be interacting mostly with things before. And so you can see this kind of a lot of things, the direction index being positive and then close to zero, and then being negative and back to zero, and a positive again, and zero and negative and zero. And so anytime you have this from negative to positive, this boundary is where you can define the TAD boundary from one triangle to the next triangle. And they can use a hidden Markov model to really identify the boundaries of, you can say, this is a chunk of plus, the red chunk, this is a green chunk, there's another red chunk, there's another green chunk. And anytime you have a green chunk next to a red chunk, that is a TAD boundary. And so in homework 5, we're going to ask you to implement a hidden Markov model to really identify the TAD boundaries. You can see, like most of the time you get this occasionally. You can see here there's some noise in the data, but overall the data should be pretty confident. And so the hidden Markov model is asking, is this the red chunk or the green chunk? And also the zero chunk. In our homework, we'll just make it easy just to state, are we mostly in the red chunk or are we mostly in the green chunk? It's similar to the coin flip. Are you in the fair coin or the bias coin, right? So only two states. So we wanted you to kind of see how hidden Markov model works. And so after they assign the TAD boundaries, you can actually look at how genes are regulated. This is one example of what people found. For example, there are 3 TADs in here. There's a little triangle to the left, which the Wnt gene resides in. There's a middle chunk. This one doesn't have a gene, but it has an important enhancer. There are some other weaker interactions. And then on the right, there's also another gene, EPHA4, and there are other interactions. And then on the right, there's another triangle with the PAX3 gene. And you can see this region is kind of interesting. The PAX3 gene somehow kind of controls the fingering and by an enhancer on the left. And so even though there are less interactions, you can see if there is a deletion in the middle chunk, this can influence whether this person have 2 thumbs or not, or you can see here. And so that gene is actually regulating the kind of the thumb side, whereas on the left side, this Wnt gene can control how many fingers you're gonna have. And so depending on whether this person has an inversion or duplication. Basically, a potential enhancer can either be interacting with too much or not enough with the Wnt gene. And this person might end up with only 3 fingers or 7 fingers. And so you can see here, because the interactions are happening within each TAD domain, whether there are deletions or amplifications or inversions, it will influence how much the nearby gene is activated. And that can change whether the person have 2 thumbs or not enough fingers or too many fingers. Right? So it's really quite fascinating. Recently, there are technology development where people are able to get this experiment working even better, and they are getting higher and higher resolution. And the new technology is called the in situ Hi-C. I mean, it's roughly the same Hi-C experiment, just doing technique-wise slightly better. They now see that on average, the TAD is around 158 kV. And so this is kind of why, for example, in our previously looking at transcription factor binding and how that influences gene expression, we usually look at all the binding within 100 to 200 kV regions, right? And they can use this better technology to see better loop conditions. You can see here on the right side, you can see this little red interaction angles. You can see these little dots. Those dots are the loops, right? And when they look at where the loops happen, especially this, what they found is that they can see a loop anchor, right? So if you have 2 things that are looping, sorry, here, they can see how the loops actually anchored at each other. And what they found is that a lot of these black dots here are enriched for a CTCF binding site. So CTCF is reported to be an interaction insulator. So when you have a CTCF binding site and there is a gene on the left and an enhancer on the right, previously it's reported that it can actually block this enhancer from regulating the gene. So it's being defined as a potential insulator. But also when people are doing more ChIP-seq, they found that cohesin is another protein complex. Cohesin binding site and CTCF ChIP-seq binding site have significant overlap in the genome. So they always co-occur in the genome. And so people were wondering what kind of chromatin interaction is happening. And a very interesting model is being proposed, and using simulation and the more experimental data, this is being considered to be a very good model. It's called a loop extrusion model. So we can imagine the Hi-C really captures the interaction average in millions of cells, but the loop extrusion model suggests that in the genome, there are these motors. So the motors are these, so the motors usually happen in pair, and these are cohesin. And they basically, once they bind to the genome, they start to kind of move, actually the motors are one direction. So you can see, if they bind in this way, they will keep going, and then occasionally they will get stuck. Whenever they get stuck is because there's potentially a CTCF binding site. But because if they are going in this direction, occasionally they do get stuck, but you don't really see chromatin interaction. If they're going in this direction, they also will get stuck in here in the CTCF binding site, but you don't see interaction. If they go in this direction, they are going further and further. Sorry. If they are going far, this direction, they are going far apart, you also don't see chromatin interaction. But if they are kind of in this way, they face each other, these two are interacting with each other, and they each kind of try to loop the chromatin in one direction. And then you can see, when they see the CTCF, they get stuck. You see the cohesin and the CTCF co-localizing. You also see chromatin interaction anchored at the CTCF binding site. And within this loop, you can see here is the loop, you see more chromatin interactions within. Okay, so basically, as long as in the genome, CTCF is binding. So that's the blue arrows here. CTCF is binding to the chromatin really tightly, and they can, and then the cohesin is this motor, and they always appear in pairs, and then they just loop. You know, every time they bind to the chromatin, they will just keep looping until a pair of CTCF stop them, and that's when they form a loop, and you will see chromatin interactions within this loop, which is quite interesting. And people have been able to use simulations and other experimental approach to really support that this type of loop extrusion really happen in the chromatin. And that's how genes within are co-regulated. In order to really visualize these data, there are a couple of a popular visualization tool. One is Drew's box, because it's Hi-C, you know, like it's kind of cute. This is developed by the lab that invented the Hi-C experiment, and so you can look at the interaction map interactively. There's another one that's developed at Harvard, Neil Scalenberg's lab, called HiGlass, and this one they have some preloaded Hi-C maps. You can zoom in and out as if it's a Google map, which is really cool, and would encourage people to take a look and see how things interact with each other. And I do see questions. Let me see. One question about the hidden Markov model. So Jack, what is your question about hidden Markov?
Yeah. Can you go back to the hidden Markov model slide? Like
Yeah, here. Yes.
Yeah, this one. Can you talk in more details about like what's the, say, like transition and what's the emission like in terms of hidden Markov model? I remember you said like it's like flipping a coin.
Mm-hmm.
So what is the fair and what is the bias coin, and
okay.
what's head and
in this
tail?
case, you can imagine the fair coin is like this region that's giving out a lot of red. The bias coin would be just, you know, fair coin or bias coin is a different hidden states. So we are trying to say this region you have mostly red, and another region you have mostly green. Well, in this case, red means if you look at the directional index, they are mostly positive, meaning that this bin, this 40 kb bin, is mostly interacting with things after it. You can see here. This is a region that have a direction. So basically, the directional index is this location, a number, next location, a number, that location, a number. And this number just tell you whether the interaction happens mostly with things after or mostly with interactions before. And if it's with after, it will be mostly a positive number. And if it's before, it's mostly a negative number. And if it's even, it will be close to 0. Right, so the transition probability is asking you how often do you change from a red block to a green block. You know, you can see roughly in the genome, we now know roughly it's 150 kb. And how often do you see a transition from a red to a green, right? Whereas the emission, you are saying, if you are in a red block, you know, how often do you see a positive number versus a negative number?
Oh, yeah. Okay, that makes sense.
And so at the end, you're just guessing, am I in a, it's like a stock market, right? If you see the stock going up, is it in a bear market or a bull market? You're just trying to call a region red and a region green. Okay, Scarlett, do you have more questions?
Yeah, I have another question.
Oh, sure.
So, basically, is that like the red and green area is all like with CTCF binding motif? Because from my knowledge, it's basically like the CTCF binding motif has the boundary directionality to decide like if it's like from the positive, from before or from after the sequence.
So what people have found is that CTCF binding sites are enriched in a region from green to red.
Oh, okay.
As a boundary. So anytime you see this triangle, CTCF binding sites are usually enriched right at the two triangles boundary.
Okay.
Yeah. And so the loop is this triangle, which means that things within this triangle are more likely to interact with each other.
Mm-hmm.
Yeah. Do we have other questions now for this tiled domains? Okay.